Today's topic is neural tube defect, and I'm Dr. Sourav Chowdhury. I'm assistant professor in the Department of Neurosurgery in All India Institute of Medical Science, Kalyani, West Bengal. So, to talk about neural tube defect, first will be the definition. So, it is nothing but a derangement of normal embryogenetic cascade during early embryonic development between the weeks of 2 and 6. So, there is a derangement of normal embryogenetic cascade. So, there are three stages by which a neural tube formation occurs. So the first stage is a gastrulation that occurs from week 2 to 3. Second stage is a primary neurulation that occurs from week 3 to 4. And third stage is secondary neurulation. These are the stages which will take us through the development of the spinal cord. And any problem with any of these stages during the development can lead to different sort of neural tube disorders or defects. So first stage is the gastrulation. So what happens is, I will take you to the photo directly. So first, after the zygote is being fertilized, there is a differentiation and then formation of something called as a morula, followed by blastocyst. So morula is a 16 to 32 cell stage and then forms the blastocyst. So this is the blastocyst, where at this stage the implantation occurs. Now, implantation is by the trophoblastic cells, the cytotrophoblast and the syncytiotrophoblast. This is the extraembryonic mesoderm. And so initial stages, there are only two layered disc inside the embryo. So what is there? It is the first layer is the epiblast, the top layer. The lower layer is the hypoblast. So it's a two layers disc, bilaminar disc it is called as. So the epiblast faces the amniotic cavity, the hypoblast faces the yolk sac. So the amniotic cavity will harbor the developing fetus in future, and the definitive yolk sac will initially give nutrition as well as blood supply to the fetus, and then in the later stages will form the gut, the genitourinary system and everything. So this is this is the cut section. You can see the hypoblast layer and then the epiblast layer. So what happens in formation of three layers from two layers? So what happens is, these layers, they are very thin. So in amidst this thin layer, there is a thickening that happens from the caudal aspect, that is from the lower aspect, which goes and elongates and goes towards the oropharyngeal membrane, that is at the top. So this elongation happens, the thickening of the cell happens, which is called as a primitive streak. So the primitive streak, this epiblast cells in the center forms a thickening which is called as a primitive streak, and it extends from the caudal towards the cranial. So the cranial most part of this primitive streak, as the primitive streak phenomenon is occurring, the cranial part of it is more thicker than the rest part of it, and that is called as a primitive node or a Hansen's node. This you can see the cranial part. This is called as a primitive node or a Hansen's node. And inside the Hansen's node, there is a furrow or a pit, which is called as a primitive pit. So what is the significance of this primitive pit? So the epiblast cells now, they jump inside this primitive pit, and the cells will enter in between the epiblast and the hypoblast layer. So something like this, you can see the epiblast cells, they enter the primitive pit and enters between these two layers. So these epiblast cells finally form the something called as a future mesoderm. So the epiblast in between, when they enter in between, they form the mesoderm. So this epiblast will form the ectoderm, then there is the mesoderm, and then there is a endoderm, that is the hypoblast cells. Okay. Now formation of this bilaminar disc to trilaminar disc is called as gastrulation. So any problem in this particular stage will give rise to issues which will involve all three layers, that is the ectoderm, endoderm and mesoderm. Now, as we have seen these epiblast cells entering in between these two layers, now these epiblast cells, as they form mesoderm, these mesodermal cells now they arrange longitudinally in a cluster on both the sides. So these mesodermal cells are yet to become something called as the notochord. So there is initially these mesodermal cells, they will arrange themselves in a longitudinal fashion, And then that particular longitudinal cluster of cells, they start cannulating. That means they form something like a tube, which is called as notochord. Okay, so we'll come here. So this tube will have a perforation in the center, and that is called as a notochordal canal. So you can see in this particular photo, you can see this epiblast. This is the epiblast. This is the hypoblast. This is the notochord. That is the mesoderm derivative. That is a notochord. All right. So this notochord formation occurs at different levels at different areas. So here you can see the notochord formation happens. Here it is incomplete formation yet. Okay, so as at different levels, as we take a cross section, you can see at this level, at this level, we are seeing an incomplete formation of the notochord. However, at this level, there is almost near complete formation of the notochord, and finally there will be a canalization of the notochord. So generally this notochord, as we have seen, epiblast cells, as the mesodermal cells goes on both the sides, the notochords generally initially are 2 in numbers. And these notochords will further more, will in future will give rise to the neural tube. So formation of notochord is very important. So gastrulation in this stage, one of the most important event is formation of the notochord. So from two notochord, there will be fusion of these two notochord and there will be formation of one notochord. So any problem with fusion of these two notochord will lead to some sort of disorder that will come across in later details. Then we are coming to the second stage, that is the primary neurulation. So here we have seen this is the epiblast cells, that is the ectoderm, the notochord, and the hypoblast cells. Now the cells in the ectoderm, on in the influence of this notochord, under the influence of the notochord, they thicken again, the ectodermal cells. Now once they thicken, they form a groove after that. From that groove, they form a fold. So from that fold, they form a tubular structure, which is called as a neural tube, which is destined to form the spinal cord in future. The, the skin formation above this neural plate occurs due to the influence of the bone morphogenic protein. So the notochord, the neural tube, once it detaches, under the influence of bone morphogenic protein, will give rise to the normal skin. Any problem in this bone morphogenic protein secretions in inadequacy can lead to loss of skin over this spinal cord. Okay. So generally this canalization of the neural tube occurs generally it was initially thought that it occurs in a zipper fashion, like we put a zip in our jacket, it occurs in a zipper fashion. But nowadays it is being found that it can occur randomly at multiple sites. The other important thing that occurs in this particular stage is something called as a disjunction. So once is primary neurulation, that is the formation of proper tunnel here. There is the spinal cord from the neural tube, and then something called as a disjunction. That is, this particular skin layer will get separated from the neural tube. That is called as a disjunction. Any problem with the disjunction will give rise to certain neural tube defects that we'll come across later. The third stage is secondary neurulation and retrogressive differentiation. So what happens, this notochordal formation that we have just spoke about, it extends till the top of the conus medullaris. If you remember, the conus medullaris is the end of the spinal cord at the level of L1 L2 junction.